In this video, we will discuss about recombinant Adeno Associated Virus Transduction Pathway. So, how this Adeno Associated Virus helps in integration of the host genome, integration of the this gene that is present within the Adeno Associated capsid to integrate with the host genome. We will understand that pathway in brief. So adeno-associated virus can interacts with the glycosylated cell surface receptor. These are the receptors that has glycans in it, and adeno-associated virus can interact with the glycans that are present on the cell surface. So after this interaction, this helps in endocytosis or internalization of this capsid with the help of this clathrin, and thereby it goes into the early endosome where, because of the reduction in the pH, there is a conformational change occurs that leads to the escape of this adeno-associated virus. Some of the adeno-associated virus goes into the nucleus, and uncoating of this capsid will happen, and some of them will go to the proteasome in the cytosol, and the peptides of these adeno-associated virus will form that might directly start its function about whatever the target genes that are present within the. So this this part will not this part will work as a activation of the immune system, as this peptide will then be goes into the extracellular matrix and will be detected by the immune system of the body, and against this antigen the immune system will generate the antibodies that can detect those antigens and kill those cells which would which have this kind of antigens present on its cell surface or within the cells. If the immune system can detect that whether a cells contains this kind of antigen, then it will try to kill the cells. Let's come to the main point. After uncoating, when the nucleocapsid or adeno-associated virus reaches the nucleus via nuclear pore entry, there is uncoating happens, and there are two kind of strategies that are used. First one is single-stranded adeno-associated virus. Second one is self-complementary adeno-associated virus. So what this single-stranded adeno-associated virus does is it has either positive strand genome or negative strand genome. Uh, so, however, for the transcription, what it needs a double-stranded. It needs to be double-stranded. So, what happens in this case is that either the DNA polymerase of the host cells will form the complementary part of this strand genome, single strand genome that is uh, that comes through this adeno-associated virus, or the nucleus will has the opposite or complementary strand uh, with, on its own. So after this strand annealing take place, what happens is that because of the virus ITR that helps in the circulation of this strand, and after that the circular monomer or circular contamer, in case there are more than one genome is present, or circular dimer in case there are two genome is present, will then be transcribed with the help of the host cell machinery or DNA polymerase as circular genome can also be transcribed by the most of the host cells, or uh, yeah, and some part of this genome will also integrate to the host genome, and the percentage is very low. That's why it's shown by the dash. And simultaneously, what happens is that, or not simultaneously, in other case, when this self-complementary adeno associated virus used, it usually comes in the double stranded form, as we can see here. So it can directly go into this circularization through this virus ITR, that helps in the circulation of this genome. And I'm not able to recall the full form of ITR. You can go through the reference that has been provided in the description. So the circular monomer will then be transcribed with the help of cellular machinery. In this case, also some part of it get integrated into the host genome. However, that percentage is very low. That's why it's shown by the dash color. So this is how the recombinant adeno-associated virus transduction pathway takes place. And this is just a brief description. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.